Well, here we are at chapter 4. I'm sure that test was not fun. Let's talk about it in class. I don't, it's mid-July. I don't even know what happened. But anyway, now we can finally start putting some atoms together and start to get closer to something that's biologically functioning and recognizable to a biologist. We've been doing chemistry, and that's great, but we need to start building things. And carbon is the glue that holds all of the atomic world together. That's basically what chapter 4 is about. I'm going to tell you, chapter 4 is a piece of cake compared to chapter 5. Chapter 5 is a beast. If you ever ask anyone who's taken this class or across the country, in Campbell Biology, chapter 5 is probably the hardest chapter in the book. And it has nothing to do with brain kind of warping toughness. It's just long. I love it because we start to then create fats, lipids, proteins, nucleic acids, the things that make up living things. I think it's great. But we got to go through chapter 4 first. We got to go over kind of the fundamentals of covalent bonding with carbon atoms and start to build bigger things. I'm going to tell you, we're going to go over this whole chapter. I'd like you to read the whole thing. We're going to go over some learning targets here. Don't write them down. We'll just go over them verbally. Definitely answer these concept check questions. I like all the pages in the chapter, and there's no websites or any of these podcast movies from Mr. Anderson in this chapter. This should be super quick. It's designed to go quickly. There's not much here in the reading guide packet. There's just a few pages. So I would say get it done. Don't get behind time-wise with chapter 4 because you want to start to go ahead in chapter 5. And a lot of chapter 5 is review also. Here's the PowerPoint. It's super short, kind of interesting and easy. So get it done quick as well as you can to get to chapter 5. Well, there will be a new Cliffs Notes assignment that I'm assigning right now and more vocabulary words for the next exam. So here's that assignment. Make sure that you keep an eye on that for the future.
To get to the heart of the global warming story, the very heart of it, you've got to look past frozen polar bears and past melting glaciers and past the changing climate. Because behind the scary news and the very scary Hollywood movies, it turns out that the scientific explanation for global warming depends to a very large degree on one particular atom, the atom that we call carbon. The more you know about this atom, the more you're going to know about global warming. First of all, it's everywhere. The lead, what we call lead in the pencil, it's not lead, it's carbon. Diamonds, carbon. You, me, yeah, you. You are mostly water, but if I squeeze you like a sponge, your dry weight, what's left of you, you're about 2/3 carbon.
Great, carbon's great, but can I have everything else back, please?
Yes, carbon is the central element of life, so much so that everything that is alive, and I mean everything, from an amoeba to a whale, from a dandelion to a redwood tree, in fact, inside every cell, in every creature bar none, you will find carbon. And why?
Yeah, why?
Because as the science writer Natalie Angier says, carbon is the duct tape of life. It stitches living things together. Carbon atoms are built to attach, to grab on to other atoms. They have, as you see, 4 points of attachment, and they love attaching. They can form jungle gyms. This is the inside of the diamond. They form sheets. This is graphite in a pencil. They form spheres. They are very social, which is why they are so suited to living things. Living things, after all, come in all kinds of shapes, and carbon can make them all. But it's carbon's special ability to grab on, to grasp other atoms that is now getting us in trouble. What starts down at the atomic level can, if we're not careful, lead to big, and I mean very big, problems.
All right, as you can tell, that was a movie on global warming. You can't get away from talking about carbon at this kind of fundamental level without talking about carbon dioxide, because it's how carbon gets introduced into biological systems. 4.1 talks about organic chemistry. Now, organic chemistry, it just means dealing with compounds which contain carbon. The hippies stole the term organic from the scientists, I guess. So please, okay, so time out. This is not a passive chapter, and neither is chapter 5. You have got to get out a piece of paper right now. We're going to draw a bunch of stuff. I'd like you to draw this. This is essentially the carbon cycle in a simplified form. And there was an essay question on this on the AP test a couple years ago. They said, oh, kids, tell us all that you know about the carbon cycle. Just understand that carbon is most readily found in carbon dioxide in our atmosphere, and plants incorporate carbon dioxide into their bodies with the help of the sun and water. We're just going to write up here water also. And you know that as photosynthesis, right? So water, sunlight and carbon dioxide come together, and some oxygen is made as a byproduct. That's great for things that need the oxygen. We need it in order to help burn this glucose, which is the base of the food pyramid. Here it is. In its ring structure, it can certainly also be in a flat linear structure, but this is just made of a bunch of carbons attached to oxygens and hydrogens. The hydrogens would be white and the oxygens would be blue. 6 carbons, 12 hydrogens, 6 oxygens. Get comfortable with this because we're going to spend a lot of time here, and I know you did in sophomore biology as well. Well, some organisms, well, everything that eats plants is looking for glucose. We burn that in a process called cellular respiration in our mitochondria in the presence of oxygen. We then create something called ATP, which helps contract our muscles, beat our heart, helps our brain to think, eyes to focus, and you name it, helps power us through our day. And we also create things like mass, structures that help the organism get bigger. And we have as a byproduct or a waste product carbon dioxide. So it's a circle, it's a cycle. What's cool is that all of this carbon that exists on our planet came from stars, from explosions of stars over the billions of years of our solar system and our universe. And so I want you to understand this. I told you the other day that you're actually not here. You're 99.99999% empty space. The actual mass that you do have can be compressed smaller than a head of a pin and weighs 140 pounds or however much you weigh. And of that, 2/3 of that is made up of carbon, but carbon comes from the stars. So you're really not here, but what is here actually is star. Congratulations. I told you before, if someone ever calls you a star, believe them. And what we then do, let's say to fuel us or to fuel our cars, is we take dead plants or dinosaurs, which are things that ate plants millions of years ago, that's been compressed under heat and pressure. It's turned into a liquid. We mine that. It's called oil. We put it in our gas tank. So really the energy for the glucose that exists in these bonds right here, it comes from the sun. And so really when you're driving down the road, what are you burning? The sun. Well, that's the same. It's the same as you walk down the hallway. You're made up of stars and you're actually burning sunlight. Isn't that awesome way to think about life? Congratulations. Let's keep going. Although cells are 90% of water, the rest of organisms consist of carbon-based compounds. You can see that here. I'll teach you what all these little angles and stuff means. That's organic chemistry. Well, this should be a picture that you have. I should have given you a couple handouts here, or maybe I didn't. Maybe I never saw you. But this is a picture that's in your book. I like this picture. It's going to show you here that that carbon likes to bond to 4 other things. So every time you see a carbon atom, make sure that you identify that there's 4 sticks coming out from it. Those sticks represent covalent bonds where they're sharing an electron on their side, the carbon side, to an electron that's bound to another atom. And we can draw a bond between that. We can also make a ball and stick model. We can draw a structural formula, which you're about to do. You name it. So go ahead, first things first, let's create the Lewis dot structures. Here's the one for carbon. Create the Lewis dot structures for these following atoms. Go ahead and press pause. And what you'll just be doing is putting dots around them, however many valence electrons they have, which are available to bind to other electrons and make covalent bonds. Well, this is what you should have gotten. Probably review by now. Good job. So then what I'd like you to do is draw the structural formula for methane, which is really fart gas, ethane, and ethylene. Go ahead and press pause and draw those. Take your time. Think about how many valence electrons there are these electrons, and then you're putting sticks, making covalent bonds, and you're sharing electrons between atoms and so on. All these are non-polar. They're going to be evenly shared. I don't see any oxygen atoms or any other electronegative atoms like nitrogen, fluoride, chlorine. What's that for now? For the electronegative atoms. Well, pretty easy for you to see that carbon can partner with many atoms other than hydrogen. It can partner with O or oxygen atoms to make CO2. We've already established that on the board. And urea, this is the stuff, it's a byproduct of protein degradation, which makes our pea yellow. Here's another segment of that movie.
All alone, there's not much a carbon atom can do. But the instant carbon grabs onto other atoms, carbons, hydrogens, oxygens, then
I'm a fuel.
that's methane.
Now watch this.
This is ethanol.
Another fuel. And check out this one. Now we're a sugar.
You're fructose, aren't you?
Let's see glucose.
No, thank you.
What about sucrose?
No.
What about French toast?
No. The point here is there's almost no limit to the kinds of structures that carbon can form. But the thing to remember is that when a carbon atom grabs onto another carbon atom, it does so with considerable force. Atoms make tight, energetic connections. That energy helps hold creatures together. In fact, carbon grips with so much energy that even when the creature dies, like this, inside the dead animal, you will still find after it's dead, carbon atoms locked tightly, closely together. And that explains why some fuels are called fossil fuels, because they come from dead things that died a long, long time ago. Though the carbon atoms inside those things are still locked, fiercely gripped inside. So there is energy in there. In fact, let me tell you a story. A long, long time ago, in a part of the world we now call the Middle East, there was once a sea, and in that sea there were little creatures called zooplankton, kind of like jellyfish. And they'd swim around all day long, filled with carbon atoms ripped inside, and then, of course, they died. Over millions of years, a lot of them died, because that's what living things do. Die. So all their corpses, lots and lots of corpses, sank to the bottom of the sea, where under pressure they gradually squeezed themselves into a mush still filled with carbon atoms, still fiercely gripped together, that we call oil. So oil then is ancient life that is liquefied, and coal is just ancient life in a rock. And in both cases, the carbon bonds that are in there tightly gripped, so there's energy inside. Now the question is, if the energy's in there, how do we get it out?
Well, I'm going to tell you how you get it out. You burn it. We burn it enzymatically and thermodynamically. All that means is stretching of bonds in our mitochondria to break these atoms to release the energy or the sunlight energy that's held in there. But you can burn it in the engine of your car. You just put spark plug or flame next to it. It also breaks the bond, releases the energy, powers the piston, which powers the axles, which turns the wheels of your car. Here are some fuels. These are called hydrocarbons, ethane, propane. You can see that we're just adding an extra carbon on these. So it's methane, ethane, propane, butane, And then pentane, penta means 5, the prefix penta, hexa means 6, septa means 7, octa means 8. But I just remember these from organic chemistry in college, the weed out class for medical students. That's what that is. Well, just understand that that there's a variation in carbon skeletons due to length. You can see here some of them are longer than others. Sorry, here's an example of a picture in your book. They're showing a fat molecule. You can see that there's different hydrocarbon chains here. Remember, hydrocarbon means many carbons and many hydrogens. Totally nonpolar, everything to the right of this fat molecule on the right side of your picture. The red dots on the left are oxygen atoms, and those would be polar, but you can see that in a fat molecule, the majority of the atoms are nonpolar. The bonds are nonpolar because of just mostly carbons and hydrogens, and that's why it doesn't dissolve in water. There's a lot of energy here in all of these long hydrocarbons. In fact, if you look at a bear that's trying to get ready for hibernation, they're really not interested in carbs because they just burn those. They want fat, and these fats are these long hydrocarbon molecules are like what fats are made up of. And then when they go in hibernation all winter, they just keep on breaking those bonds as they're hibernating, and that just provides enough energy to keep them alive. But you can start to see these variations here. You can see branching as a variation. I'm going to tell you that there's the same number of hydrogen and carbon atoms in each of these molecules, and we'll get to what that's called in a moment. You can see here differences in double bonds placement. You can see 1-butene is off the first carbon, and 2-butene has the double bond off of the second carbon. Notice when you get a double bond, remember carbon can only bind to 4 things, and so it's no longer bound to 2 hydrogens. If it's bound in a long chain with other carbons, it can only be bound to 1 hydrogen right at the double bonders. That's because, go ahead and count the carbons there. The double bonds are going to take up 2 electrons for each carbon, so it only has 2 left. See that? I wish I could point that out. This here, hydrocarbons can undergo reactions that release a large amount of energy, that's for sure. You can also see them in ring form. Often we see them in ring form in biological systems because they do contain some oxygen atoms, and it's just because of polarity that they'll ring up. And that's why when I showed you earlier, the glucose, it's in a ring form. Sometimes it can be straight, but it's most often in that form. Well, I've written up here, fat is hydrophobic. It's nonpolar, generally speaking, because of all those atoms that are sharing electrons equally. It doesn't dissolve in water, you know that, and it's called a hydrophobic molecule.
In the last episode, we learned that carbon atoms form bonds and connections inside living things, like, say, a tree. Now, if you cut down a tree, you can make the tree into lumber. And then you can take the lumber and make lumber into pieces of paper, like this piece of paper. So if there are carbon bonds tightly gripped, if there's still energy inside this piece of paper, how do I get the energy out? Inner valve. The fire excites atoms in the paper so they heat up and they jiggle. When they jiggle, they break apart. And then immediately they find new partners. And these new partnerships are generally more efficient and more stable, so that leaves a little energy left over. That's why it's warm when you're near a fire. It's the energy being released from those carbon atoms changing partners. And there's another way to break a carbon bond. In a parrot. The parrot has carbon locked inside, but when the carbon gets down to my stomach, there's no fire in here, but there are digestive juices which break those carbon bonds, which once again release energy, and all of that energy takes the form of calories, and then those calories power me. The same thing happens when you gun an engine. You spark the gasoline, which excites the carbon atoms, breaks their bonds, releases energy, and off you go. So we break carbon bonds all the time. We just don't realize it.
I think thought.
You think thought.
In fact, one could say that human civilization began when the first cave person
That not thought.
figured out how to make a fire
Some thought.
or how to break carbon bonds.
What a
And those fires over the centuries became furnaces, which powered engines, which lit up cities, and created towers and vast highway systems filled with cars. It turns out the more people become rich on Earth, the more we use carbon. Carbon is the atom that equals success. Although, of course, there are other atoms that produce energy. There's always uranium.
All right, so let's stop there. Sorry about the shirtless moment in my cartoon. Let's talk about isomers. Now, isomers are compounds that have the same number of atoms, just often in different arrangements. You can see that right here on my handout. This is a picture from your textbook. There's three different types. We'll talk about structural, geometric, and enantiomers. Let's talk about structural isomers first. We touched on this earlier with the branching. So the idea is that they have the same number of atoms, but just in a different arrangement, and that's going to cause definitely different properties. Maybe their melting point will be higher, their freezing point or boiling point will be different. They'll fit inside of a little kind of notch in an organism a little bit differently. So it's really important, the shape of a molecule. It says here, structural isomers have different covalent arrangements of their atoms. That's for sure. Well, there's something next called geometric isomers. I want you to find this. This is really important in biology. There's two different types. There's cis and trans isomers. Cis means the same. And in this instance, the idea is that the majority of the molecule is on the same side. So I'm going to press pause here. Let's go up here first. I want you to draw this. So whenever we see these squiggly lines like this, what we're representing here are carbons. Every time you see a kink, you can imagine that that's a carbon. And then you could fill it with as many hydrogens as that carbon can hold. So this one on the end is going to have 3 available electrons to bond to hydrogens, and this is going to have 2, and so on. We can go down this, but remember when we get to the double bond here, that these guys will only be able to bind with 1 hydrogen, and so on. So this is just kind of a lazy man's way to build a structure of a long hydrocarbon chain. So here's another one. We can draw this here, and you could imagine all of these carbons and hydrogens on here. And then once we get to the double bond, I'm going to put a hydrogen here. Sorry, that's a hydrogen. I'm going to put another one here. And you can see in this instance, Aaron, that the majority of this compound molecule is on opposite sides structurally of the double bond. You see that? So the hydrogens are on opposite sides of the double bond structurally, and the majority of the rest of the compound is also on opposite sides. Very important, because structure equals function. So the first one that I have up there on the screen that you have on your screen is when the majority of the molecule is on the same side. That's called cis. And when it's trans, the majority of the molecule are on opposite sides of the double bond. That's very important. We'll get into this in chapter 5 a little bit more. Make sure you take a moment and understand that. Last type of isomer is called an enantiomer. And what these are basically are mirror images of one another. So go ahead and make sure that you take a look here. And so if you imagine if you had your hand and it was facing towards a mirror, let's imagine that this board is a mirror. The mirror image would look like this, right? These are called enantiomers of the palms of my hand, the mirror image, because if you were to try to place them on one another, they are not symmetrical. They would not fully fit. They're symmetrical when they're facing like this, but if you were to take them and say, oh, they're the same thing, well, they're not the same thing. And you can see that here. I wish I could point up at the board. try to take the L isomer, L meaning left in this instance, and then try to flip that around so you can put that over the top of the right one. And what you'll find is that you can't. What it's showing you is that there are different atoms that are going into the screen and that the CH3 is coming out of the screen, and they just don't fit over one another. They're called enantiomers. There's the L isomer or the D isomer, which is derecha, which I think is in Spanish means right. Oh, and you know what right is right or correct, also right. If you ever go to like a famous museum, you'll see it in all the paintings. The bad guys have like a knife in their left hand, and the angels are holding like food in their right hand. Or whatever. Anyway, L and D here. There are mirror images or these enantiomers in our body, like these different types of dopamine. You can see these are mirror images of one another, these molecules, but they actually do entirely different things in the body. You can see that. So read about enantiomers in the body. There's one final thing that we have to talk about, and that's called functional groups. The whole idea is that we can get these long hydrocarbon chains, that's great, but they don't do anything really by themselves. We have to there's 7 different groups that we can put on there, and these are combinations normally of oxygens and hydrogens. You can see here a small number of chemical groups are the key to the functioning of biological molecules. You can see way off to the right side of those top figures where it says CH216. That means that there's 16 carbons and hydrogens just in a squiggly line all by themselves, and then on the left side there's some sort of combination of oxygens there, and that's going to give the whole molecule its characteristic in living systems. So distinctive properties of organic molecules depend not only on the carbon skeletons, but also on the molecular components attached to them, for sure. See here these different arrangements of oxygen atoms. So here's an example. Your book is going to talk about these 2 sex hormones, estrogen, which is found primarily in women, but men make it also, and testosterone. Press pause and count how many atoms are different between estrogen and testosterone. And there are many atoms present here. I forgot the number. I meant to look it up before the lecture. It's like something in the 40s or 50s atoms. And there's just a few that are different. But those few atoms that are different can lead to tremendous differences in the organisms in which they work. So the point is that structure equals function. It's really important. It's not a coincidence or haphazard what the structure is.
Dear gentlemen, science has a confession. Your life actually began as a female. Well, phenotypically or physically speaking, that is, though some prefer to call this stage gender neutral. As you might know, when 2 people mate, they each combine half of their genetic information together, 23 chromosomes each. One pair of these chromosomes helps to determine your sex. If you're female, you contain 2 X chromosomes, one from your mother and one from your father, while males have 1 X and 1 Y chromosome. At the moment of conception, you were, of course, male or female based on your genes, but the Y chromosome doesn't kick in right away. In fact, the first 5 to 6 weeks of embryonic development are attributed to the X chromosome alone, and females grow from embryo to fully developed through the influence of only the X chromosome. So what does the Y chromosome do? Once activated, one of the genes on the Y chromosome, known as the SRY gene, has 2 functions: to inhibit certain features of the X chromosome, and to impose through dominance the physiological traits that a male must have. The ovaries descend and become the testes, and the labia fuses to create the scrotum. If you're a male, take a look. Though it's more pronounced in some men than others, you'll likely notice a line or ridge of tissue on the scrotum, which is called the scrotal raphe. The skin of the scrotum and penis are zipped up like a zipper as the layers of cells develop in the embryo. And the male variant of the clitoris? Yep, that's the penis. If the Y chromosome does not become activated for some reason, then the female phenotype or physical appearance will persist in a male. While we're at it, this is also part of the reason males have nipples. The nipples form before the activation of the Y chromosome and SRY gene, and thus remain through development and life. But you don't develop breasts. Sorry. Got it, Vernon?
I remember working and doing a preceptorship with a pediatrician once, and he told me that 10% of male babies that when they're born have undescended testicles, which just means that they have ovaries, which are going to produce eggs. They've been biologically, chemically changed into testicles, and they need to get down to the scrotal sac. And so all guys have a tube, and so do women. I don't know why the ladies have it, to have the ovaries descend down into the scrotal sac. And it's just a kind of a weaker spot in the abdomen, and it's an area that can be torn easily if someone's lifting something. And a hernia is created when a tear occurs in the abdominal wall, and then small intestine will wiggle its way in there over time, and then it will descend. And I had another doctor tell me once that he worked in an inner city, and he said that he would sometimes once a month there would be a man that would come in and as pale as a ghost with a trench coat late at night and say, have a big problem, doc. And the doc would say, okay, show me. And the guy would open up his coat. And I heard this from a surgeon here at Vail Health. And he just said what would happen is they would have torn their abdominal wall. Small intestine would have gotten in that tube and through gravity descended into the testicle. And so they would have to have surgery because you can't have that food tube twist, or it's called torsion, and block up, or else you'll get septic and die. So you can imagine 1,000 years ago, the Egyptians making the pyramids or whatever, and these young men, super strong, lifting these big pieces of marble or whatever, stone, and tearing their abdominal wall in that weak spot and so on. So that's a hernia for you. Well, there are 7 functional groups that you need to know about. And there's a handout that I hopefully have given to you. The number and arrangement of these functional groups gives each molecule its unique property for sure. Here are the 7 functional groups. Do you need to memorize these? And the answer is yes. You need to know these names, and I'll show you exactly what you need to know with each of these. Your textbook does a good job explaining these, but I just don't like the picture in your textbook, and I'll tell you about that in a second. Here's a movie that might help you.
There are 7 functional groups. Yes, 7 functional groups to know. Hydroxyl, OH, carboxyl, COOH, amino, NH2, 4, NH3, so hydro, SH, carbonyl, CO, methyl, CH3, and phosphates, OPO3. Hydroxyl, OH, hydroxyl, OH, they're alcohols like ethanol, ethanol, hydroxyl, COH, carboxyl, COH, yes, yes, that's sugar, it's what it's sour.
Okay, make it stop. Sorry. Okay, well, you can watch that on your own. If you're humming that during the exam, I'll know why. Again, I don't like this picture. The old handout is better than the new one. I have it here. Hopefully, I'll get it to you. I'll get it to you somehow. I just think this one's a little bit too summative. I'm going to show you what you need to know for each of these. So go ahead and find and follow along. For amino group, you don't need to memorize all this. You just need to circle where it says NH2 up there in the structure. 2 hydrogen atoms bound to a nitrogen atom. I would also go down to the functional properties at the bottom where it says it can pick up an H+ from surrounding solution, and sometimes it can be ionized. That's it. That's all you need to know. If you rewind this, you'll know what you need to know for the test. Just recognize it. It's going to be a large component of amino acids. They'll all have an amino group on the end. You'll see that. Next one, sulfhydryl group. I would say it's just SH. And something that's important is if you go to example where it says cysteine is an important sulfur-containing amino acid, that's important. Circle the 2 bullet points down there under functional properties. 2 sulfhydryl groups can react, forming a covalent bond. This cross-linking helps stabilize protein structure. You'll see that when we study proteins in chapter 5. And then actually you don't really need to know about, that's just an example of the second bullet point there for sulfhydryl group. The next one, phosphate group. We see this often in ATP has phosphates, also on the backbones of nucleic acids like RNA or DNA. We see phosphate groups. It's OPO4, so I guess it's OPO3 negative, sorry. I would say, I would circle up there where it says structure and all that language that's up there. Under example, it says, takes part in many important chemical reactions. Glycerol phosphate provides the backbone for phospholipids. Again, we see it in DNA and RNA, as well as ATP. That's it. Don't worry about the functional properties down there below. Next one, methyl group. It's a CH3. We're going to see this sometimes DNA gets methylated in order to turn on or off genes, which allows the genes to be expressed or not expressed, which has huge effects on an organism. You can see that under 5-methylcytidine down there under example. It just says that it's a component of DNA that has been modified. Go ahead and circle both those bullet points. You'll also see methylation occur in differences of those sex hormones that we saw earlier. Flip it over. Carboxyl group is C double bond OOH. We see carboxyl groups a lot. So much that I forgot it's July, okay? Oh dear. I would just underline under functional properties where it says has acidic properties, and you're just going to see carboxyl groups. They exist also in amino acids before they come together. You'll see that. Oh, we're almost done here. Hydroxyl group, OH. I would say something that's important with the hydroxyl group I've written up here is that it's polar, and you find it in alcohols. Finally, carbonyl group, a C double bond O. If you go down your handout, underline where it says ketones and aldehydes, and then at the bottom under functional properties, the second bullet point says these two groups are also found in sugars, giving rise to two major groups of sugar. So you're going to see these carbonyls, and they're around. We've already seen them a little bit earlier today. Well, in case you needed more, just kidding. Okay, we're done. Thanks for paying attention to Chapter 4, everybody.